Hello, I'm Cassandra Liu from Diocesan Girls School, and in this video I will be sharing my project, investigating an alternative to Agarose for DNA Gel Electrophoresis with you. Before I talk about the details of my project, here's some background information. You may have heard of the term DNA fingerprinting before, and it actually involves a common and important analytical technique in molecular biology called DNA Gel Electrophoresis. In DNA Gel Electrophoresis, DNA fragments in a sample are separated according to their size when an electric current is passed through a piece of gel in which the samples are loaded. As DNA Gel Electrophoresis allows us to accurately compare the DNA of individuals, some of its uses include identifying criminals based on DNA collected on a crime scene and paternity tests. Usually, agarose gel is used in DNA gel electrophoresis as it is easy to prepare, firm, strong, clear, non-toxic, and electrostatically neutral. However, it is also expensive and hence not readily available in developing countries. With the aim of increasing the accessibility of DNA gel electrophoresis, I investigated whether there are any more readily available alternatives to agarose gel. Due to the objective of this project, the gels that I tested could all be made with affordable materials purchased from a typical supermarket: gelatin gel and tapioca starch gel. You may have heard of gelatin before, as it is commonly used to make jelly. I chose to try using it as it is first of all clear and second of all can be prepared easily. The second gel that I tested was tapioca starch gel. You may have also heard of tapioca starch because it is used to make boba pearls. I chose tapioca starch because other types of starch have been used for protein electrophoresis in the past, and investigated as alternatives to agarose in DNA gel electrophoresis. Moreover, tapioca starch gel is a relatively clear starch gel and has never been used in DNA gel electrophoresis before. Preparing the agarose gel was simple. I put together a protocol from multiple sources online, as I mentioned before, DNA gel electrophoresis is a common analytical technique. As for the gelatin gel, I followed the same procedures as preparing agarose gel, as they were similar to how people usually prepare jelly anyway. The dissolving portion of the procedures were fine, but the gel didn't set at room temperature as expected, so I put it in the fridge for a few hours until it was reasonably firm. Preparing the tapioca starch gel was the most difficult. I first attempted to use the same procedures as preparing agarose gel, but upon microwaving the mixture of tapioca starch and TAE buffer, the mixture separated into two phases: a small clump of highly viscous and sticky solid and a runny liquid. The solid was not a suitable gel, and the runny liquid could not gel, so I tried heating the mixture on a hot plate while stirring with a magnetic stirrer instead. A milky, homogeneous mixture was formed, but the tapioca starch did not dissolve completely in the TAE buffer. Therefore, I microwaved it for short periods of time with vigorous stirring with a glass rod afterwards, and the tapioca starch finally dissolved. Similar to gelatin gel, it only set after it was placed in a refrigerator for 2 hours. In fact, it was so gooey that it didn't appear to have set, but as the substance stayed intact, despite sticking to my fingers when I touched its surface, technically it has gelled already. After preparing the gels, I conducted DNA gel electrophoresis with each of them, loading the same samples, using the same voltage on the gels for the same duration. In short, I came to the conclusion that both gelatin gel and tapioca starch gel are unfortunately inappropriate for DNA gel electrophoresis, especially in comparison with agarose gel. Gelatin gel was too fragile, while tapioca starch gel lacked structural integrity, was sticky and gooey, and fluoresced under UV light such that DNA bands could not be observed. Although the gels that I experimented with cannot replace agarose gel, there are still plenty of other more accessible gels that have the potential to do so, such as other starch gels, modified starch gels, and pectin gel. After conducting these experiments, a conclusion that I have come to, though, is that a good gel for DNA gel electrophoresis definitely has to be firm and strong. It was really cool to be able to see and try using apparatus in a chemical and biomolecular engineering lab. Though there were small accidents as I made my way through my experiments, I've definitely learned a lot in the experience. Thanks to Mr. Tsuedo and Mr. Francis Falk, who supervised my experiments, I was also able to catch a glimpse of what studying science and conducting research at a university level is like, while waiting for my gels to set. I learned a lot from the rest of my project as well. It was nerve-wracking at times, and I doubted whether I would be able to finish it at all, as the techniques and knowledge seemed so out of reach to me. In the end, I couldn't try some of the things that I may have wanted to, because of my limited knowledge of laboratory techniques and time constraints, but I'm really glad that I pushed through and was able to try a few gels, because I was genuinely able to learn and experience so much. Thank you ABFYE, EDB, Professor Marshall Liu, Kelvin, and Natalie for bringing me this wonderful experience. And thank you for listening.